Yes. Remember that we went to the eye part, and in addition, I remind you that in addition to the images that you have here, you should go to the first lecture that you have it in Priya's presentation, Introduction to Histology, and there is a slide there that is taken from a retina preparation that is actually an immunohistochemistry with an antibody to KIF3A, which is a kinesin. And it shows you how you can identify in which layer the immunolabeling is if you put it side by side with a retina section that's stained with hematoxylin and eosin. But let's go fast to the ear. The ear, this is a very nice drawing, and it shows you how it is organized. All the action is present in the inner ear, and in fact in two parts of the inner ear. One contains the vestibule and the semicircular canals. The vestibule contains the utricle and the sacral, and the semicircular canals are in a different region than the vestibule. And the other part is the cochlea. And this is because the ear, the inner ear, has two functions. It senses the equilibrium and the motion, and the cochlea, it's the auditory part that senses the vibration caused by the sound. And when I tell you the equilibrium, and it senses the motion, the rotational motion or the directional motion and the accelerations, it's not the motion of the body, it's the motion of the head. If I rotate my head, or if the head moves in and accelerate in a certain direction. And that is sensed by the vestibular apparatus. The cochlea is the auditory apparatus. And each of them it's set into action by an event that comes into the inner ear. I told you already the equilibrium and the motion of the head is sensed in the vestibular apparatus. So this is caused by the motion of the head, the sensing of what's going on in the cochlea. It's the sensing of the vibration caused by the sound that comes through from the external ear, inner ear, middle ear. So what's happening in the middle ear, it's restricted to the auditory part in the inner ear. And now I also want to tell you that the nerves that then send to the brain the signals that are perceived in the inner ear as action potentials is the cranial nerve 8, the vestibular branch of the cranial nerve 8, and the cochlear branch, auditory branch of the cranial nerve 8. So I will read briefly the text that's written here. Is from the Hunchera textbook. So the acoustic meatus, which is here, so this is the external ear, it's all elastic cartilage. The acoustic meatus of the external ear ends at the tympanic membrane. This is the tympanic membrane, and its mucosa contains sebaceous and ceruminous glands that produce an antimicrobial substance, the cerumen. And these ceruminous glands are of the same type as the what was called the apocrine sweat glands, but they in that case they also secreted by merocrine secretion. But these ceruminous glands, they have a role in fighting bacterial infections. So what happens next? The tympanic, the middle ear, it's this one here. It's also known under the name the tympanic cavity. It's the tympanic cavity of the middle ear, opens to the nasopharynx via the auditory tube or Eustachian tube. And it has an important role in equalizing the pressures, that when the sound wave creates pressure here, it needs to be equalized at some point, and that occurs with this auditory tube. Within the tympanic cavity, an articulated series of 3 small bony ossicles, the malleus, the incus, and the stapes. So this is the largest one, malleus, incus, stapes. The stapes, it's I think the smallest bone in our organism. And it connects the tympanic membrane with the oval window in the wall of the internal ear. Now, when the whole of the inner ear is only for the acoustic part, the sound wave comes and creates pressure on the tympanic membrane. The tympanic membrane vibrates and sets in motion the ossicles, the stapes, the incus, and the malleus. And the malleus, it's here at what's called the oval window and creates the pressure into the inner ear, and that pressure then distributes into the cochlea, and this is how the sound wave penetrates into the inner ear and generates the motility of the hair cells that are present in the cochlea. And the hair cells are neuroepithelial cells, actually. We'll see soon. So the internal ear consists of a membranous labyrinth containing endolymph. The membranous labyrinth is enclosed by the temporal bone's bony labyrinth, which contains perilymph. This is not easy to understand, and I had a lot of trouble finding the best way to explain it to you. So you should understand that all this apparatus, the audiovestibular apparatus, is carved into bone. They are carved into bone. And there are spaces that are carved into bone and channels that are carved into bone. And into those spaces, which are filled with a liquid that is called perilymph, there are other structures in the form of either ducts or chambers that are inside that bony labyrinth. And those are called the membranous labyrinth, not because they are surrounded by a lipid bilayer, but they are surrounded by soft tissue. So what you should think of the organization of the inner ear is, I would start better with the membranous labyrinth. And you see here the biggest part here in the vestibule, it's this sac-like structure. It's a large sac. It contains inside a fluid that is the endolymph. And it has extensions that run through these semicircular canals, and they are called semicircular ducts. You should think of this as a canal in which a pipe is extended. The pipe would be the membranous labyrinth, and the channel would be the bony labyrinth. The channel is filled with perilymph, but the pipe inside is filled with endolymph. And you should think of this in the same way with what's happening here in the cochlea. The cochlea, it's a structure, it's like a stairway that narrows as it goes upwards. And it is carved in bone, but a middle section in that carving, it's bounded by soft tissue. The section that's bounded by soft tissue, that is the membranous labyrinth. What's bounded on the outside by bone, that is the bony labyrinth. And if you would do a section here, you would see the bony labyrinth, all these bluish structures, they are surrounded by bone, directly by bone, and they contain the perilymph. However, there is one of them that is bounded by soft tissue that contains cells, and that contains the endolymph. In the cochlea, there is that stairway that contains actually two channels that are bounded by bone, and they contain perilymph. But in between them, there is another one that is bounded by soft tissue, and that is the membranous labyrinth. And the key to understanding of how the equilibrium is sensed, the motion is sensed, and the sound is sensed is the fact that there are cells, neuroepithelial cells, that are embedded in the membrane of that membranous labyrinth, and they stick with their apical region inside the membranous labyrinth, but have the connection to neurons on the other side. So here, the vestibule, it's present in a cavity that's surrounded by perilymph. The cells are present in these thickenings, and there are several thickenings. The ampullae are present at the entrance into each of these semicircular canals, and the maculae are present, the thickenings, one in the utricle. The vestibule, it contains this utricle that has a thickening here that is part of the vestibular apparatus, and also the saccule that appears to connect, and in fact it connects to the auditory apparatus. It's also part of the vestibular apparatus. And the thickenings here are the maculae. So the membranous labyrinth has a central vestibule with two subdivisions. The utricle connects to the three semicircular canals, and the saccule connects to the cochlear duct. The walls of the utricle and saccule each have a second area, the macula, which contains both sensory hair cells with synaptic connections to sensory nerves and supporting cells. These mechanoreceptors monitor static position and linear acceleration. And you should think of this like this is the apical portion. The hair cells are the ones that are stained darker in violet. All these other are supporting cells, and the yellow structures are actually these are the dendrites of the neurons that form the vestibular branch of the cranial nerve 8. This structure, you should imagine that these apical regions reach inside the vestibule. You don't see them because they are inside, and they stick the stereocilia and the kinocilium inside into the endolymph in the vestibule. Now, there in the inside, what actually is present, it's what's called the autolytic membrane. It's made of proteoglycans. They swell in water, and this contains calcium carbonate particles. The ear sand that people talk about, the ear sand, is there. And now the stereocilia and the kinocilia are very stiff, and they initiate an action potential that's transmitted to the brain when the calcium carbonate particles, the otoliths, touch them. And they will touch them when motion will occur and move this entire autolytic membrane. That will start oscillating, and the calcium carbonate particles will hit the stereocilia and kinocilium. When will that happen? When we move, when we start moving from a static position. And that also senses the acceleration. Remember, if I accelerate, that gel-like substance will move into the endolymph, and that's what triggers an action potential. Now, what's happening here in the semicircular canals? Each of the semicircular ducts, oriented 90 degrees from one another, has a terminal ampulla. You should remember this name, macula and ampulla and sacule and utricle. So has a terminal ampulla region with a, yes, it's recording, with a thickened crista ampullaris containing hair cells that contact a gel-like cupola. So these are the hair cells. These neuroepithelial cells, the sensors of motion, are hair cells, and they are called hair cells because of the stereocilia and the kinocilium, which stick out like hair. And there are such hair cells present in the ampullae, and they stick their stereocilia and kinocilium again in what's called crista ampullaris, in the cupola, that also contains this a gellose matrix that will move. Actually, it's not a gellose matrix, but there it's endolymph in those. The hair cells stick with their apical region, with the hair, towards the interior of those ducts. So what will make them move? If we rotate the head, the fluid in all those three semicircular ducts will move, and it will move in different directions, and it will stimulate the generation of action potentials from these hair cells. But look how ingenious this system it's done. So we live in a space that is three-dimensional. So, and rotation occurs in this three-dimensional space. How would you sense the three-dimensional rotation? You put these semicircular ducts aligned with the three dimensions, and this is how they are there. And in this way, we know exactly how our head moves and rotates. This is the key on how we sense, and this they detect the rotational movement. I will not go through this text. I tried to explain the best way I can how this happens. I listened to a talk by Howard Hudspeth at the neuroscience meeting in November. He actually talked about the cochlea, but the mechanism is pretty much similar as with the vestibular apparatus. But he is the one who who identified these hair cells, who identified how they work, and he is such a great speaker. You should have listened to his talk. I tried to get it. I'm not sure if I can. They removed it from the Society of Neuroscience website. Now, let's switch to the cochlear auditory apparatus. So I told you that the cochlea, as you see it here, it's sort like, imagine it as a cone. To that cone runs a stairway, and at the bottom, the stairway, it's quite wide, but as we go towards the top, it becomes narrower and narrower. And here you see structures here, structures here, 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 and here. You should understand that they are actually part of the same continuous structure. And that continuous structure, it's also it's the organ of Corti. And what you see here repeats here, and you see it here again and again and again and again, because it is actually a continuum. The same structures that you see in one place, they repeated themselves throughout this entire cochlea. And if you, this is a two-dimensional structure explaining what you would see in the cochlea. So there are three channels, I should say, and two of them are part of the bony labyrinth, and the middle one is part of, it's actually the membranous labyrinth. And the two parts of the bony labyrinth are the scala vestibuli and the scala tympani. The middle one is the scala media or the cochlear duct. And they are drawn like this here in two dimensions, but they really go up to the top region, which is the helicotrema. At the helicotrema, the scala vestibuli and scala tympani, they fuse with each other. And the scala vestibuli and scala tympani contain the perilymph. The scala media or cochlear duct contains the endolymph. And the sound wave comes through the oval window, it sets in motion the fluid, the perilymph in the scala vestibuli, that sets in motion the fluid in the cochlear duct, that sets in motion the so-called basilar membrane on which the hair cells are attached, and they move, and they have stereocilia that are embedded not in that otolytic membrane, but also in a gellose structure, the tectorial membrane. And now these are from the textbook, and this is what I just told you. And try to envision that this entire membranous and bony labyrinth are like a spiral going upwards. And this is why what's present there, the organ of Corti, it's called also the spiral organ of Corti. And now let's see the organization. So you have here the bony labyrinth, the scala vestibuli and scala tympani, and the membranous labyrinth, it's the cochlear duct or scala media. This is bone surrounding, but here this is soft tissue. This is soft tissue. This is also soft tissue. It contains some cells, and this is the spiral organ of Corti. And this spiral organ of Corti, it's easy to understand if you understood the hair cells in the vestibular apparatus, in the maculae and in the ampullae. So this would be the Reisner membrane that it's at the place where the scala vestibuli meets with the cochlear duct. Here it's the basilar membrane, and on this basilar membrane are attached with their base part the outer hair cells. These are here are 3 rows of outer hair cells and the inner hair cells. The most important ones that will signal to the brain the auditory signals and will generate the action potentials are the inner hair cells. And they make a synapse with the neurons from the of the cochlear nerve, and the cell bodies of those neurons is in this spiral ganglion. It's a ganglion. It's in the peripheral nervous system. So when the sound wave sets in motion the Reisner membrane, the endolymph that's present in the scala media will set in motion the basilar membrane. It will move the outer hair cells and the inner hair cells that have their cilia stuck into this tectorial membrane. And that will trigger the action potential, especially from the inner hair cells, also to some extent from the outer hair cells. And this is how the sound is detected. What's actually happening is that sounds of various frequencies will set in motion various regions in this scala vestibuli according to the frequency. And so it will generate action potentials from hair cells at various levels along those spiral organ of Corti. You should know the organization of the cochlea with the, you should know which is the scala vestibuli, which is the scala tympani, which is the Reisner membrane, the basilar membrane, outer hair cells, inner hair cells, and you should also know what are the cells in this region that is more intensely stained. That is the stria vascularis, and it has some, it's the vascularized strip. It contains epithelial cells that will secrete the endolymph. They secrete the endolymph here, which is different than the perilymph in the other 2 chambers. And they take the endolymph in the end, like all fluids, they originate from blood. So this is highly vascularized region, and this stria vascularis produces the endolymph. This is the textbook image. This is from the virtual microscope. So it's so well set. And again, this is just a section, but you should imagine that all these structures, they run upwards and in a spiral until they reach the helicotrema, where the scala vestibuli and scala tympani will meet. This is a drawing showing the inner hair cells, outer hair cells, tectorial membrane, basilar membrane. I think that this is it. If you go to virtual microscope assignments, this is something that will almost certainly come up in the exam, this image of the eye. And also, you should know that which the position of the utricle and saccule, that they are present in the vestibule. You should know that this structure is the cochlear nerve. You should know the names of all these structures, because you will see this image, you will see numbers on it, and then you will see a list of structures, and you will have to put the corresponding number next to it. I think that this is everything that I wanted to tell you. I still may want to go to Canvas. I think that last Thursday I showed you, but I want to make sure. Okay. So we go to our spring 2024, and you don't see it now if you log in, but the exams will be TBL1 quiz, the Word file and the PDF file in the folder February 1 TBL1 quiz. It's not published, so you don't see it, but you have all the documents that you need to prepare in the February 1, February 7 module. You download this, you answer the questions, and you send them to me by email, to me and Priya. And the time allowed, it's 55 minutes. If you are almost done and you would still need a few more minutes, ask me, send me an email and ask me, can I have a few more minutes? Of course, not 15 minutes, not half hour, but a few more minutes I will always give you. And I'm sure that you will do fine at the quiz. You will have to score in this quiz, and then I will make the final score will be the average of your score and the maximum score, which is 5 points that you automatically earned for the second quiz. So it's easy, and it's just 5 points. And it's 5 points. It will tell you how the exams are here. And I'm sure you'll do well. Thank you very much. Again, it lasts longer, but these are long lectures. We talk about the two tissues, and the second part we'll talk about only one, and we'll also do the preparation. I wish you success and good night now, and have then a restful weekend.
Thank you, Professor. Good night.
You're welcome. So is ear and eye on midterm? No. And again, we won't have a Zoom meeting next Thursday. We won't meet in class. It's just you download the exams from Canvas at 6. I probably will place them there at 5 minutes before 6 or so. And then you send them to me via email. And don't forget to also add Priya. And I have uploaded now Priya's PA review sessions, everything till the midterm exam. And she has also two sessions in preparation for the TBL, and listen to them. Some of you told me that they are very helpful, and I'm sure that they are very. Have a good evening.